Okay, good afternoon or good morning, depending on when you're listening to this, I guess. This is the podcast for the Molecular Genetics Lecture Regulation of Eukaryotic Gene Expression. So hopefully this will, like the other podcast lectures, provide a reasonable resource when you come to revise for this module. So we've got a lot to cover in this lecture. More than I would like. However, there's a limited number of lectures that we can deliver to you on the module, and for that reason I've had to try and condense what is essentially almost a module's worth of information in some ways into a single lecture. So I'm going to be very explicit about what the learning objectives are for this lecture in the next slide, because what I don't want to do is instill panic in people. I don't want people to look at this lecture and think, there's no way on earth I can learn all this material, there's no way on earth I'm going to be able to understand all this material, and therefore I'm not going to engage with this material. That would be a big mistake. One of the reasons why this is such an important lecture on the module is that this entire module forms a basis for your understanding of the level 3 module, Medical Genetics. And much of what we're going to talk about for the next 50 minutes is covered in a lot more detail in the next academic year. So it really is in your best interest to try and just get your head around some of this content. I'm going to be going through things at quite a superficial level, and as we'll see when we look at the learning objectives, I really want you to be able to develop, at this stage, develop your understanding of these kind of things, rather than end up with a really detailed and broad understanding of these topics. That will come in medical genetics next year. So what we're going to be talking about then? Well, essentially we're talking about gene expression in eukaryotic systems, from the very beginning of gene expression, if you like, which involves the actual remodeling of the chromatin, the chromosomal DNA, if you like, to enable gene expression to begin, through to how the act of gene expression itself is regulated and proceeds, through to what happens following gene expression with regards to mRNA processing and translation of the mRNA. And I guess it's really this part here and the modification of DNA structure which are vastly different from what you'll have covered in the prokaryotic lectures with Dr. Ross. In particular, the post-transcriptional and post-translational level of regulation in eukaryotes with regards to gene expression is much higher. So it's not simply a case of turning on gene expression, making a gene product, and that gene product going away and doing a function in the cell. There's a level of fine-tuning that's present in eukaryotic systems that's just not there in prokaryotic organisms. So the learning objectives then are really, as I've just mentioned, to start developing your understanding of these sorts of aspects of molecular biology. So I won't expect you for the exam to really have any further information on any of these topics above and beyond what we're going to talk about for the next 50 minutes. In other words, you need to develop a broad understanding of how these different mechanisms operate, and just try and start to get your head around how these things fit together to tell the story of gene expression in eukaryotic cells, indeed in our cells, in human cells. So that's broken down here into 4 things. Understanding how histone proteins regulate gene expression or modifications to those proteins. Understand how eukaryotic transcription occurs. So this is, remember, DNA transcription is the copying of the DNA information into an mRNA molecule. mRNA processing is a very complex and important stage of gene expression in eukaryotic cells. You need to have a broad understanding of what that is and how the different events contribute to gene expression. And finally, you should have a reasonable understanding of how mRNA translation is regulated in human cells also. So the first thing to say, which is to state the obvious really, is that gene expression in eukaryotic systems is far more complex than in prokaryotic organisms. So if we look at this figure here, this is a cartoon of what goes on in, for example, E. coli or a prokaryotic cell in regards to gene expression. Effectively, it's very simple. You've got a couple of conserved sequences here in the promoter of the DNA, which is just upstream of a gene. The -35 and -10 refer to the number of base pairs upstream of the initiator codon, the start codon for the gene. And what happens is, just upstream of where the gene coding region begins, you get recruitment of RNA polymerase via a factor called sigma factor. Sigma factor is a basic transcription factor, so it promotes transcription promotes gene expression. RNA polymerase is the enzyme that copies the DNA into RNA, yeah, which is obviously a fundamental step in gene expression. There are some other elements which can be involved, upstream regulators which can activate gene expression, but generally it's a relatively straightforward system. Now, obviously there are levels of regulation above and beyond just how this physically works to turn on gene expression. You've been learning about operon systems, etc. from Dr. Ross, but the actual nuts and bolts, that, if you like, the machinery that does the actual DNA transcription itself is quite straightforward. Unfortunately for you guys who have to try and understand this in the next 18 months, the same's not true for eukaryotic cells, for human cells, where the level of regulation of the machinery that copies DNA into RNA is much, much higher. It needs to be higher because if it isn't, we don't have the fine-tuning ability, the very exquisite regulation that's required to produce an organism as complex as we are. We need a more complex machine in order to achieve a more complex goal. So you don't need to fret too much about this figure. It's there to make a point, but the constituents of it we are going to talk about over the next few slides. Just a few things to point out. This is a promoter region here. The Tata box is a part of that promoter sequence, and these things here are all transcription factors. Lots and lots and lots of them. Embedded within them is the RNA polymerase, which is called RNA polymerase 2 in humans, in eukaryotic cells. The real difference comes with regards to two things. The first is the fact that our DNA is not just flying around inside the cytoplasm like bacterial DNA is, maybe associated with one or two proteins. Ours is highly organised into high order structures. In other words, the DNA is wrapped around these so-called histone octamers, which we're going to talk about in a moment. It's wrapped around histone proteins to form the so-called beads on a string structure. So you have to deal with this, the fact that your DNA is not accessible. So how does the cell do that? And the other thing to note is that we also require this complex called a mediator complex, which I'm not going to go into too much detail about today, other than to mention it's required. And the mediator complex effectively links upstream regulatory sequences to the actual RNA polymerase complex here, which is associated at the gene promoter. So, having said how complicated it is, the actual, again, physical act of copying DNA into RNA is very, very similar, almost identical in eukaryotic cells as it is in prokaryotic cells, insofar as there are three stages. There's the initiation stage of transcription, which is where the RNA polymerase enzyme leaves the promoter and starts actually copying the DNA information into mRNA. There's the elongation stage, so this is when the RNA polymerase fires off properly and starts copying the DNA sequence into RNA at a relatively high rate. And then there's the end of transcription, so this is a point at which termination of transcription occurs. So the RNA polymerase reaches the end of the gene, recognises the sequence, and then stops making RNA. So initiation, elongation, and termination, three processes from E. coli that also ring true in humans. So let's start by talking about chromatin remodeling then. So this figure here basically shows this beads on a string structure. You should have seen this now already in previous modules. I think we mentioned it briefly in human genetics. You should certainly cover it in more detail in cell biology, I would imagine. Effectively, it illustrates how our DNA is organised inside our nuclei. And that is that the DNA is wrapped around these so-called histone octamers or nucleosomes. So what nucleosomes are 8 independent histone proteins associated in the shape of effectively like a football or golf ball or sphere. And around that sphere the DNA is wrapped 2 times in such a way that the actual space that DNA then takes up in the cell is reduced. So that's the first structure that's formed. Once you've wrapped the DNA around these nucleosomes, the nucleosomes themselves then kind of assemble and wrap around one another to form this so-called 30 nanometer filament, or the solenoid. And these 30 nanometer filaments again form higher order structures, and that's what you see in the actual chromosome. So if you look at a chromosome spread and you see the hazy, fuzzy outline of the chromosome arms, what you're actually looking at are these solenoids. structures that form the actual high order structure of the DNA in your cells. So the presence of these structures, these nucleosomes, has a profound effect on gene expression because, to put it simply, when your DNA is wrapped around one of these things, all those proteins that are required to actually transcribe the DNA, the RNA polymerase II, all the associated transcription factors, the mediator complex, that big group of proteins simply can't get access to the DNA to copy the information that's there. So in order to get access to the DNA, these things need to be remodeled. They need to be modified in such a way that the DNA that's wrapped around the histone is freed up, so that it is accessible then to RNA polymerase II and all the transcription factors. So how does the cell do that? Well, it utilises a whole family of proteins which are termed nucleosome remodeling complexes. And what these protein complexes do effectively is they manoeuvre the nucleosomes around the genome in such a way that the DNA that's wrapped around the nucleosome is made accessible for gene transcription. A major player in this is the remodeling complex known as SWI/SNF, which is an ATP-dependent complex that effectively shifts around these nucleosomes, revealing parts of the DNA sequence which the gene wants to transcribe. So let's consider the figure here. Yeah. What you can actually do is recruit complexes such as SWI/SNF, which will then start modifying these nucleosomes, twisting them so that the DNA that's wrapped around them moves around and becomes accessible. So it's easiest to explain this, I think, by this figure. So you see this red region here. So if that's the promoter sequence of a gene that you want to express, in its current state here, wrapped around the nucleosome, all the transcription factors that are required to kick off gene expression are unable to access that DNA sequence. When a chromatin remodeling complex enzyme comes in, such as SWI/SNF, it's able to manoeuvre this DNA in such a way that it's no longer wrapped around the nucleosome. It's kind of pulled along to expose it. And once it's exposed in this internucleosomal region, this region between the two nucleosomes, the necessary transcription factors can then associate with the DNA and turn on gene expression. So it's, in theory, I would strongly urge that you try and apply this logic to all molecular biology mechanisms that you might cover on the rest of the degree. If you think about it in logical, simple terms, about what you need to achieve in order to express genes when you have DNA that's wrapped around these sorts of nucleosome structures, it actually makes complete sense and it's relatively easy to understand. All you need to do in order to express a gene is start moving the DNA to a point where it's in between these two nucleosomes so that you can get access to it. All the SWI/SNF and other nucleosome remodeling complex enzymes do is to achieve that. All they do is they manoeuvre the DNA so that you can get access to it with the transcription factors, and once that happens, gene expression can occur. Now there's a second independent way that the cell can actually promote expression of genes via remodeling of these nucleosome structures, and that's via modification of the histone proteins themselves. So this is a lateral view of a histone octamer. This shows the DNA that's wrapped around the histone octamer, the double helix of the DNA, and these structures inside here are the actual histone proteins. So as I've mentioned, there are 8 histone proteins, hence the name histone octamers, and each of the histone proteins possesses at its N-terminal end its so-called tail region. So as you can see there, the bulk of the histone proteins is tightly associated in the inside of the nucleosome, but for each of the histone proteins there's this tail region that kind of protrudes through and outside the DNA. So in other words, the tail region is kind of outside that nucleosome ball, and therefore it's very accessible to enzymes which might want to modify that tail. And indeed that's what goes on, that's how another layer of regulation is introduced into this system. So by modifying the actual amino acids that are present in these histone tails, the cell can cause the histones to either grab more tightly or more loosely to the DNA. So for example, if you modify a histone in such a way that it starts to lose its grip on the DNA, then that DNA sequence might become more accessible to gene expression factors like transcription factors, etc. If you modify the tail in such a way that it tightens its grip on the DNA, gene expression is less likely to occur. You repress gene expression. So it's another way of actually altering gene expression levels at a very exquisite fine-tuning kind of level. It tends to be certain amino acids that get modified on the histones, and they include lysines, arginines, threonines and serines, and different types of post-translational modifications occur to each of these amino acid residues, including acetylation, methylation, phosphorylation, and other ones as well now. These tend to be reversible effects. In other words, if the cell chooses to activate gene expression by deacetylating a histone, it can then repress gene expression by reacetylating it at a later time. It's not a one direction kind of system. And something you might want to have a look at, if you want to do a little bit of extra reading on this, is the concept that there might be a histone code, and that's the idea that the actual histone proteins themselves might regulate in some way the genes that are expressed in different cell types. And the histone code refers to the actual pattern of these modifiable amino acids that appear in the tails of certain histones that are associated at certain parts of the genome. So just to recap then, what we've been talking about in regards to histone modification, nucleosome remodeling. It's this concept that the fact that our DNA is wrapped tightly around these nucleosomes prevents gene expression from occurring. So in order to activate gene expression, we need to remodel this chromatin structure. And the remodeling of chromatin, the remodeling of this DNA nucleosome structure, is a major component of eukaryotic gene expression. Generally happens in an ATP-dependent manner via complex enzymes such as SWI/SNF. And by complex, I mean more than one component. It can also, however, occur via modification of the actual tail region of the histone proteins themselves. Remember, it's the histones that make up that sphere, the histone octamer. And the point of both mechanisms, the point of SWI/SNF and modifying histone tails, is to effectively expose regions of DNA that we wish to express by moving the nucleosomes out of the way. In a nutshell, that's what's going on. But exposing the DNA to what exactly and for what purpose? Well, that's what we're going to talk about next. What happens once you've got a piece of DNA exposed and ready to be expressed? So we've already said that transcription in eukaryotes is a lot more complex than in bacteria. It's exemplified initially by the fact that we have 3 different RNA polymerases in humans. Bacteria have just the one. Try and emblazon this in your mind now. It's RNA polymerase 2 that's responsible for making messenger RNA in humans and indeed in eukaryotic cells. And this is a really huge, complex molecular machine, half a megadalton in size, 500 kilodaltons. So this is a crystal structure which is Roger Kornberg solved from Stanford for RNA polymerase 2. I think he got a Nobel Prize for this. It's a massively complex enzyme, RNA polymerase 2. As you'll see, it's, in addition to actually just making the mRNA, it's kind of almost like a regulatory scaffold protein. So this is a huge kind of machine to which other regulatory proteins latch on during transcription. So it doesn't just make RNA, this thing, it's also responsible for regulating the entire system too. However, considering how complex it is and how clever it is, it's unable to actually make mRNA synthesis all by itself. It can't initiate RNA synthesis alone. In order to do that, it needs to associate with something called the pre-initiation complex, which it itself is part of. Yeah. So this entire system, this setting up of this pre-initiation complex, this large group of proteins, which include the RNA polymerase 2 enzyme, is an analogous system to that occurs in E. coli, where sigma factor recruits RNA polymerase, so that the RNA polymerase efficiently recognises and binds to the promoter region of the gene. So you can think of that process, sigma factor recognising the promoter and then recruiting RNA polymerase. The analogous system in us is RNA polymerase 2 associating with this pre-initiation complex at the promoter of a gene in order to get ready for gene expression to begin, transcription to begin. So what other factors are required in this pre-initiation complex other than RNA polymerase 2? Well, these are the so-called general transcription factors, and these general transcription factors are required in order for RNA polymerase 2 to copy DNA in vitro. So in other words, if you were to try and do an experiment, which you can do relatively easily, whereby you have a gene you wish to express in the test tube, in a tube in the lab, you can add RNA polymerase 2 enzyme to the tube, you can add your DNA sequence of interest you want to transcribe, but if you want the RNA polymerase II to actually start copying the DNA into mRNA, you also have to supply a whole range of these general transcription factors to the experiment. If you do that, then the RNA polymerase II and the transcription factors will associate and start making mRNA from your DNA template. Now, the situation in actual cells, in vivo, is even more complex, because in vivo you need an additional set of factors as well, including things like we've already talked about, nucleosome remodeling proteins, and also this mediator complex, which I mentioned at the top of the lecture, that we'll talk about in more detail next year. So for the rest of this lecture on transcription, I'm just going to focus on the actual nuts and bolts of this pre-initiation complex, to try and give you an idea about how that assembles at the promoter of a gene. So what we've got here is a eukaryotic promoter sequence. This is what's called a canonical promoter, which basically means it's your vanilla bog standard promoter that you'll see in front of most genes in the genome, and it has several important elements. This is what's termed the core promoter region, and as you can see, there's a -35 and -25 elements here, which are similar to what occurs in bacteria. You've got your transcriptional start site here, which is your initiator element, and then sometimes you actually get regulatory elements that are downstream of the core promoter also. All these extra different domains effectively perform a similar function to, again, what sigma factors doing in E. coli. These different domains are responsible for recruiting transcription factors, which then recruit RNA polymerase II to the correct part of the genome, in other words, just upstream of a gene you want to express, in order that transcription can begin. So I've listed here the different components of the pre-initiation complex and the order in which they start binding to the promoter sequence. Very simple list of facts. If you can try and remember the order these things are recruited and a rough idea about what each thing does, then that's pretty good, to be honest. So again, we'll go into this in more detail next year. So what occurs then before gene expression begins in human cells? Well, the first thing to occur is actually a protein called TATA binding protein, which recognises and binds to this TATA box region of the promoter, which you'll notice on the last slide. The TATA binding protein is actually part of a larger multi-protein complex called TF2D. So it's this TF2D protein complex which is the first thing to arrive at the actual human promoter before gene expression begins. And it arrives by virtue of TATA binding protein recognising and binding that TATA box. Once TF2D is associated with the promoter, it then recruits TF2A, which comes in and really locks down TATA binding protein then. So the association of TATA binding protein initially with that TATA region is relatively weak. However, once TF2A is also recruited, that binding becomes stabilised. So you've got TF2D, then TF2A. Once TF2A is recruited, TF2B is recruited. And what that does, in addition to binding TATA binding protein, is it forms a link between the large TF2D multi-protein complex and RNA polymerase II. So it's kind of a bridging protein, TF2B. Once TF2B is there and it can form this link, then we see recruitment of TF2F and RNA polymerase II. And these things arrive at the promoter together and associate with this TF2D, TBF2B complex, stabilising it further. So I appreciate that there's a lot of letters and numbers flying around here and it's very difficult to remember all these terms. All I can say is that you need to try and remember them. It might help to think about putting them in context. All this TF refers to transcription factor. So transcription factor 2, the 2 refers to the fact it's a transcription factor for RNA polymerase II. So if you remember that we're talking about transcription factors for RNA polymerase II, the only thing you need to remember really are the actual letters here. So D, A, B, F, E and H. Now, if you can think up, I can't remember the correct term for it, but some kind of memory device for remembering the order of this, that's fine. So where are we? We've recruited several of the pre-initiation complex by virtue of TATA binding protein binding the TATA box, TF2A stabilising that, TF2B forming a link, which allows the recruitment of TF2F and RNA polymerase II. Once you have RNA polymerase II recruited, you get recruitment of TF2E and TF2H. So TF2E binds this growing pre-initiation complex and then itself recruits this really huge multifunctional TF2H complex. And it's TF2H that actually is responsible for separating the DNA strands apart and kicking off the elongation stage of transcription. Incidentally, if anybody who likes to link lectures together, which I'd encourage you to do, TF2H also contains these nucleotide excision repair components for transcription coupled repair, which we talked about in the last lecture. So that's, once you get TF2H recruited, that's the pre-initiation complex formed on the promoter of the gene. So once you've formed the pre-initiation complex, what needs to happen then is we need to kickstart transcription. So you need to be able to tell the RNA polymerase II, everything's in place, start copying the DNA into RNA. So how's that achieved? Well, this escape's orchestrated via the C-terminal region of RNA polymerase II itself. And the C-terminal region of RNA polymerase II was a special name, it's called the carboxy-terminal domain, CTD of RNA pol II. And it's a really interesting part of the RNA polymerase II protein that's unique to eukaryotic polymerases. It's a huge, of huge importance to transcription and also transcriptional regulation, as exemplified by the fact that if you start comparing the C-terminal domains from different organisms, the more complex the organism, the larger the C-terminal domain. So in each case, the CTD is made up of these heptapeptide repeats, but the number of repeats that are present increases as the complexity of the organism increases. So in yeast, which is the most simple eukaryote, there's 27 repeats, in us there's 52. So one of the ways that this C-terminal domain can regulate everything is via modification of the C-terminal domain amino acids by phosphorylation. And that's what happens in order to kickstart the elongation stage of transcription. So we've recruited the pre-initiation complex, of which RNA polymerase II is a component, to the promoter of a gene we wish to express. Now we need to tell RNA polymerase II to start copying the DNA into RNA. How is that achieved? Well, it's achieved because one of the components of this large TF2H complex is actually a kinase protein. So remember, kinases are proteins that phosphorylate other proteins. And what this kinase does is it phosphorylates part of the C-terminal domain of RNA polymerase II, telling it then that it's ready to start transcription and kick-starting copying the DNA into RNA. So it's quite a clever system, because only when you actually recruit the last component of the pre-initiation complex does the RNA polymerase II enzyme get told it's ready to start copying DNA into RNA. So you can't start making RNA before the entire pre-initiation complex is present. So it's a nice self-regulatory mechanism. So once that phosphorylation of C-terminal domain occurs, the entire much of the pre-initiation complex actually starts to disassemble. A lot of those transcription factors are lost, and RNA polymerase II kind of escapes and sets about the business of copying the DNA into RNA. In order to do this, it requires a few elongation factors to be recruited, and again, it's the C-terminal domain of RNA polymerase II that does the recruitment of these elongation factors. So as you may be aware, when you actually kickstart transcription, it doesn't just go from not transcribing DNA to fully transcribing DNA immediately. There's kind of a stalling process whereby RNA polymerase II will copy one or two nucleotides and then stop. So it starts the process, but it won't start it proper until it receives a second signal, which tells it, right, everything's go, and then things kick off properly. So that initial stuttering start is triggered by phosphorylation of the C-terminal domain via a kinase in TF2H. The actual big go signal is given by another kinase called PTEF-B. And this only gets recruited following disassembly of many of the general transcription factors. Briefly mentioned TF2S, which is something I've not talked about. This promotes elongation by reducing pausing of RNA polymerase II. So TF2S kind of just expedites the entire system. But I don't want you to worry too much about that. What I want you to worry about is the order in which those transcription factors are recruited, and then particularly what job they're doing. The fact that TF2H is required to kickstart transcription by phosphorylating the C-terminal domain of RNA polymerase II. One final thing to talk about then, once we've got elongation underway, is this problem about nucleosomes. So we've made the promoter region of a gene accessible by modifying DNA histone interactions, like we talked about at the beginning of the lecture. But what happens when you start copying the DNA and you reach another histone that's present in the actual chromatin itself? How does the RNA polymerase enzyme actually get around the fact that much of the DNA it needs to transcribe is wrapped around these histone proteins? Well, it does that via a protein complex called FACT, which stands for Facilitation of Chromatin Transcription. And this is a complex of proteins which kind of just races ahead of the RNA polymerase enzyme, removing chunks of the nucleosomes in such a way that the actual RNA polymerase II enzyme can pass through the nucleosome and around it to copy the DNA sequence into RNA. The clever thing is that once RNA polymerase has gone past the nucleosome and copied that information, FACT then reassembles the nucleosome so that it's intact again, making sure that there's no, therefore, permanent damage to chromatin structure. So it's kind of paving the way for gene transcription to occur by modifying the histones, these nucleosome regions, ahead of the actual RNA polymerase II enzyme. So what happens then when RNA polymerase II reaches the end of a gene that you're expressing? How does it stop? The current model for explaining transcriptional termination in eukaryotes is the torpedo model, which is nice and manly. I'm sure it was a man who came up with this. It works by quite a clever mechanism that I'll tell you about now, effectively by virtue of the fact that once RNA polymerase II passes a sequence in the DNA, in the gene, which tells it, right, okay, that's the end of the gene, the mRNA it's been copying will be released, but the RNA polymerase II enzyme actually still carries on copying an RNA molecule. However, there's an important difference in this RNA molecule compared to the one which corresponds to the gene that's been expressed, insofar that this RNA molecule lacks a cap, and we'll talk about caps in a moment on mRNA. So the fact that this mRNA lacks a cap allows the cell to recognise it as foreign, as incorrect, and what occurs then is that a very, very active, highly processive RNA polymerase enzyme called XRN1, or RAT1 in yeast, which is a much better name, binds to this uncapped RNA and chews it up. And it chews it up with such speed that when the enzyme reaches the RNA polymerase, it kind of launches it, torpedoes it off of the DNA, terminating transcription. So this is what's going on here. This is gene transcription underway. This poly-A site tells the RNA polymerase that it's the end of the gene, and also triggers cleavage of the mRNA that's been produced. So this is your mRNA molecule that will go on to be translated. These proteins cleave the mRNA to release it. So this is a released mRNA that then will get taken into the cytoplasm for translation. RNA polymerase II carries on making an RNA, but this RNA lacks this M7G, which is a cap region of the RNA. So XRN1 recognises it, chews it up, and launches the polymerase II enzyme off of the DNA, terminating transcription. So just to finish the transcription section of the lecture, to say there are obviously other RNA polymerases in eukaryotes. I alluded to that at the beginning of the lecture. These are RNA polymerase 1 and 3, and I've listed what these RNA polymerases are responsible for here. Some of them require general transcription factors to operate, and where that's the case, I've listed that also. You don't need to worry about these RNA polymerases other than to know that they exist and that they're involved generally in the production of small RNAs, tRNAs, etc. That's enough. The RNA polymerase you need to understand a lot more about is RNA polymerase 2, which is what we've been talking about. So a quick recap again, because obviously we're covering a lot here. We've already said the structure of the genome greatly affects gene expression, and that you need to remodel that in order for gene expression to occur. I've told you that RNA polymerase 2 is the enzyme responsible for transcribing all mRNA in our cells, and that it requires a large number of accessory factors, this so-called pre-initiation complex, and also this mediator complex, which you don't need to worry about other than to know it's required in order for gene expression to occur. The C-terminal domain of RNA polymerase 2 is a regulatory region that's essential for regulating gene expression in humans, and it regulates it via phosphorylation, which is carried out by other proteins within the system. So, for example, TF2H contains a kinase which phosphorylates this C-terminal domain to allow RNA polymerase 2 to begin transcription. Transcriptional termination is performed by an RNase enzyme called XRM1 that kind of torpedoes RNA polymerase II off of the DNA once the gene's been transcribed. So that's a complex system there, and I'm sure listening to it again, even following the lecture, it's not completely clear. All I can do is suggest that you try and listen to this and do some wider reading until you start to understand the basic processes that are in place. I know there's a lot of names to remember. Remembering the names isn't as important as understanding the basic logical processes which take place in order for gene expression to occur in eukaryotic cells. So what happens to these mRNA molecules that are released then by RNA polymerase II following transcription? Well, I've already said that mRNA processing is a very important aspect of gene expression in humans, and I'm going to tell you why now, briefly. So, in contrast to what goes on in prokaryotes, where translation and transcription are effectively coupled. So in other words, when an mRNA emerges from RNA polymerase, ribosomes associate with that mRNA almost immediately and start making it into protein. That doesn't occur in eukaryotic cells. In eukaryotic cells, the site of transcription, i.e. the nucleus, is separated by a membrane from the site of translation, i.e. the cytoplasm, where the ribosomes exist. So the RNA has to go on a journey from the nucleus into the cytoplasm. And to survive that journey, and indeed to regulate its translation, eukaryotic mRNAs undergo several modifications. So the major three modifications that actually occur are listed here. So the first is that to the 5' end of the mRNA, a methyl cap, a modified methyl cap is placed. The primary function of this cap we'll discuss in a second, but effectively it protects the mRNA from degradation from RNase enzymes. You should be aware by now that when all our genes, or certainly 99% of our genes, contain intronic sequences, these are non-coding sequences which need to be removed prior to gene expression, prior to translation, of the mRNA molecule. And finally, the 3' end of the mRNA also needs to be protected from degradation, and that's achieved via the addition of a so-called poly-A tail. So the cell adds a long stretch of adenine nucleotides, A's, to the end of the mRNA molecule in order to protect it. Polyadenylation. And that's summarised in this figure here. So what do these different modifications actually do? Well, the 5-methylguanine cap, as I've just said, protects the mRNA from degradation. And this occurs more or less immediately. As soon as the mRNA emerges from the RNA polymerase II enzyme, this cap is placed on the 5' end. And the cap itself is a modified methylated guanine nucleotide. And it's modified in such a way that the RNases can no longer attack it. Well, again, cover that in more detail next year, but it's important you know that it happens and why it happens. That's the kind of knowledge I want to instill in you in this lecture. The introns then. The introns need to be removed. All eukaryotic organisms utilise introns in their genes. How are these non-coding regions removed from mRNA? They're removed by a massively complex and huge enzyme called the spliceosome. So the spliceosome you can think of as an RNA protein molecular machine. In other words, it's got hundreds of different proteins in it, dozens of different RNA molecules which have got catalytic activity, and together these things come together to effectively recognise where these intronic sequences lie and remove them from the mRNA, so that you end up with a mature RNA molecule ready for translation into a protein. We'll go into splicing in a lot more detail next year. Finally, there's a modification to the 3' end of the RNA. So once the RNA polymerase reaches the end of the gene and the RNA molecule gets cleaved, the 3' end is immediately modified so that a poly-A tail is added. Lots and lots, up to 200 of these adenine nucleotides. And this is important for protecting the RNA, but it's also important to regulate translational efficiency, so to regulate the translation of the RNA into protein. And we'll talk more about that in a second. So that's the modifications which immediately occur to the RNA to enable it not to be chewed up and destroyed and to get it in a mature state ready for translation so that the introns are removed. But how does it get from the nucleus into the cytoplasm? So it's, as I've said, it needs to go on a journey from that one region of the cell to the other. Well, this isn't just a passive process. The RNA just doesn't diffuse out of the nucleus. It's actually taken out of the nucleus by a whole complex of proteins. The most important of which is this protein TAP P15. So TAP P15 is what's known as a nuclear export factor, and this protein binds to the mRNA and then via an association with the nuclear pore complexes, which lie in the nuclear membrane, drags the mRNA out of the nucleus and into the cytoplasm. Interestingly, mRNA always leaves the nucleus 5' end first, cap end first. So we've known this for a number of years because of microscopy studies. We now have a molecular explanation for this, and that is that the TAP protein is recruited exclusively to the 5' end of the mRNA via an association with the cap. It's a little more complex than that, but in a nutshell, that's how it works. So you have this directed export of mRNA in a 5' to 3' manner through the nuclear pore complex via an association of the mRNA with this nuclear export factor TAP. Don't worry about all the other proteins on this figure, they're not important. They're just there for detail. The thing I want you to remember is that TAP's involved in export, and the way it works is it drives export of the mRNA through the nuclear pore complex 5' end first. So just a quick recap on mRNA modification then. Capping, splicing and polyadenylation, they're the golden triangle, the three you need to remember. The 5' end of the mRNA is capped to protect it. The 3' end has a poly A tail to protect it, and any introns are removed via splicing. Now splicing, as I've said, is very complex. I'm not going to talk about the mechanism, but I want to talk about alternative splicing briefly. And this is a concept that if you have a gene with 3 introns in, there are different ways in which you can splice that gene. The first way is to remove all 3 introns. The second way is to remove 2 and leave 1 in. Now if you do that, leave that intronic material present, you're obviously going to produce a different gene sequence, a different mRNA sequence, for translation. So this is how the cell is able to get more bang for its buck from the number of genes which are contained in the genome. And this explains a phenomenon by where the Human Genome Project revealed to us that we as humans have around 22,000 genes, which is the same number as what are present in worms, C. elegans, which is a model organism, which we'll talk about later on in the module. How on earth can we have a similar number of genes to a simple nematode worm, yet be so much more complex biologically? Well, largely the answer lies in this alternative splicing mechanism, and the fact that whereas a worm will use 1 gene to make 1 protein, we will use 1 gene to make many proteins, in some cases many thousands of proteins from a single gene that are subtly different, and that's achieved largely by this alternative splicing mechanism that we'll discuss next year in medical genetics. So this is a figure that kind of just shows mRNA processing in its entirety, the capping and the removal of introns, the polyadenylation, the export into the cytoplasm, and then the translation of the mRNA by the ribosomes here. And that's what we're going to talk about next. So you should be aware, because you get a lot of microbiology on the degree course, that ribosome translation in prokaryotes initially occurs via the recruitment of the ribosome via something called the Shine-Delgado sequence. So this is a particular sequence which exists in the DNA to which the ribosome binds, because it recognises it and it binds. Now it's not the same in eukaryotic cells. We don't have a Shine-Delgado sequence. So how do we recruit ribosomes to mRNAs for translation? Well, in a nutshell, this process is achieved via the 5' methyl cap, which we talked about earlier on, and a number of accessory proteins called eukaryotic initiation factors. Initiation relating to initiation of translation. So I'm going to go through this simply. You don't need to obsess over the detail, you just need to understand. One of the important things to understand is that it's cap dependent, that you need this 5' methyl cap to recruit ribosomes in order to translate eukaryotic RNA. But to give you a little bit more detail about what's exactly going on, there's these figures here which I'll talk through now. So this is the mRNA as it comes out of the nucleus into the cytoplasm. It's got a 5' methyl cap upstream of the actual start codon here, the ATG, or AUG obviously as it appears in mRNA. Remember, thiamines in RNA are replaced by uracils. What occurs following export into the cytoplasm is this 5' end of the RNA gets modified. This methyl caps associates with a group of proteins, the most important of which is this EIF4E, this little protein here, and I'll tell you why it's important later on. And these proteins bind then to the 5' end of the mRNA and this is the start of what we call the 48S pre-initiation complex. The second part of that complex is the readying of the actual small subunit of the ribosome. And by readying, I mean the actual initiator codon, so the initiator tRNA, the methionine, gets associated with the P site of the small subunit in the ribosome, so that it's ready to do that initial step of transfer once it starts translating the DNA sequence. And there are several other accessory proteins associated with it, but it's really the recruitment of the initiator tRNA to the P site that I want you to focus on here. So those two things together form effectively this so-called 48S pre-initiation complex. So this is the modified mRNA with the EIF4E protein and the cap, and this readied small subunit of the ribosome with the initiator tRNA present here. Once you get formation of this 48S pre-initiation complex, many of the actual proteins dissociate from it, and you get a new protein called eukaryotic initiation factor 5b, which then recruits the large ribosomal subunit, the 60S subunit, to form a mature ribosomal subunit which is termed 80S in eukaryotes, not 70 like in prokaryotes. So now what we have is the initiator tRNA at the P site, the ribosome's wrapped around the mRNA that we're going to translate. There's a vacant A site ready to accept the next tRNA, and effectively translation can get underway. So you get transfer of the initiator codon, and then you start getting addition of the subsequent amino acids to that initiator codon as translation proceeds. So this process is called cap-dependent translation, so called because it depends on the actual intact structure of the mRNA cap and this EIF4E protein. There are other systems that exist in eukaryotes that don't require a cap. Many viruses utilise systems which use something called an IRES, which stands for internal ribosome entry site. And what these are specific sequences of DNA to which a ribosome will bind in the absence of a cap. You don't need to know about those other than they exist. So we've managed to assemble this mature ribosome on the RNA. It's started to actually translate the RNA sequence, which the mechanism of which is more or less identical to the mechanism in bacteria, which is why I'm not going into it here. What I do want you to understand is that in eukaryotes, translation is not a one-shot affair. So a ribosome doesn't bind to the cap, move along the RNA making a protein molecule, and then fall off. Yeah, it's important to understand that conceptually. What actually happens is this. The RNA molecule, which has a cap and a tail, forms a loop structure. So it forms a closed circle structure. And this is achieved by the virtue of an interaction between this EIF4E protein, this protein I said was important earlier on, and this poly-A binding protein, a protein that binds to this poly-A tail. And these two proteins come together via an interaction with a third protein, EIF4G. So these three things together enable the RNA to form like what's called a closed circle structure. So what actually happens when a ribosome is recruited is the ribosome recruits here, copies the RNA to give a protein, and then it will do the same again, and it'll just go round and round and round and round. And this process of continuous translation is what's known as steady state translation of RNA. The important thing to realise is that the point of regulation for this steady state translation is this protein here, EIF4E. So the cell can modify EIF4E's ability to bind to this protein, and by doing so modify translation. So think about what would happen if the cell decided to modify EIF4E in such a way it was no longer able to interact with this protein, no longer able to form the closed loop structure. The upshot of that would be vastly reduced translational efficiency, less gene expression. Yeah? Conversely, if the cell promotes this kind of structure to form, it upregulates gene expression. So what we've got is yet another layer, another point at which gene expression can be regulated in human cells. And that's why EIF4E is often referred to as the master regulator of translation, and it's also why EIF4E expression is often upregulated in many cancers. So if you accidentally end up with a situation where this EIF4E protein is really always promoting these closed circle structures, really ramping up the amount of protein being produced, that can lead to cellular transformation and cancer. Unregulated expression of proteins such as proto-oncogenes, for example. The final part of gene expression is the degradation of the mRNA itself, and that's a very important aspect as well. mRNA is an unstable molecule. We need it to be unstable because we want to be able to turn off gene expression as quickly as we can turn it on. There's no point producing an mRNA molecule that will sit around in a cell for weeks and weeks getting made into protein. If suddenly the environmental situation changes and a cell decides, right, we need to cease making this protein now, failure to do so will be dangerous for the cell. mRNA is inherently unstable, so that when gene expression stops in the nucleus, any mRNAs which are still kicking around in the cytoplasm will be there for 1 or 2 hours, maybe a little longer, and then they'll be degraded and gene expression will cease. There's another system I just want to alert you to as well, which is interesting, called nonsense-mediated decay. And this is a system which effectively exerts quality control on the mRNA that's been exported. I've written what this is here. Have a look at it if you want. However, I'll deem this as extra reading, this nonsense-mediated decay. You're not going to be asked any questions on this in the actual exam. I just put it in there as a mechanism of interest that you might want to engage with some wider reading after the lecture. So to recap then about translation, once transcription is finished, mRNA undergoes several processing events. It's capped, it's given a tail, it also is spliced to remove introns, and then it's exported into the nucleus, into the cytoplasm where translation occurs. Translation elongation, the actual process of translation, is very similar to prokaryotes. The thing that's different is that ribosomes are recruited in a cap-dependent manner, and that once they're recruited, they don't just copy one mRNA and fall off. They form part of this closed loop structure that's able to then really efficiently produce a lot of protein using just a continual cycling of ribosomes around the mRNA. Bad mRNA, such as those containing nonsense mutations, can be degraded via this process called nonsense-mediated decay that you may wish to have a look into. We'll go into it in more detail where you will need to know this next year, so you might wish to engage with that wider reading now to prepare yourself. But you don't need to know about nonsense-mediated decay for the exam for this module. So relax, there's a lot of information there. I appreciate that that's a little bit overwhelming. I hope, however, it has given you a good, broad, solid introduction to gene expression in eukaryotic cells. You're going to need to understand these processes in order to do well in the medical genetics core module at level 3. So it's really worth your while listening to the podcast, doing a little bit of extra reading and trying to just understand some of the fundamentals about the processes which we've talked about this morning. This is a useful link here to an open source website which has several nice animations explaining transcription and translation. I'll put some extra reading on Blackboard for this, and the molecular biology of the cell and molecular biology of the gene to a lesser extent are excellent core textbooks to which you can refer for some additional reading and alternative takes on what we've been talking about here. Okay, thank you for your attention.